Good evening, guys. I'm just gonna check whether you can hear me. If you don't mind clicking. Oh, perfect. Thank you. Okay, so today we are on our last lecture, lecture number 9, and we are going to be talking about high performance liquid chromatography or HPLC. So the things that we're going to cover this week are basic principles of HPLC, and then we'll talk about different HPLC separation mechanisms and the application of each of them in biopharma, so for bioanalytics and the analysis of biopharmaceuticals. And we are on the last column, well, last row in our table here. So we are talking about chromatography, which is same as mass spec, extremely important method that can give us a lot of information. So almost all of the information that we get from mass spec, but it's more user-friendly, let's say, than mass spec, not as expensive, and we don't need operators that are as highly trained. They still need to be highly trained, but not as complex as for mass spectrometry, so it's used in all stages, and it's a really popular method to use that is used in QC labs. So first we'll just go through overview and introduction of chromatography. So chromatography in general can be used on large scale and small scale as well. So we are using chromatography in manufacturing for purification. So the main aim of chromatography on the manufacturing scale is to purify our molecule of interest. So we use different modes of chromatography in order to remove things that we are not interested in. So things like other proteins that are coming from host cells, toxins, aggregates, viruses, DNA, so anything that we don't want there. On a small scale, in a QC lab, this is where we use chromatography to characterize our protein to just ensure that production that has been done produce the protein with the correct sequence or folding or structure, that we have correct purity and post-translational modification. So it just allows us to look at our product and make sure that our product is what it's supposed to be. So chromatography is a separation method used to separate, identify, but also quantitate and analyze complex mixtures. And as you said, can be preparative or analytical. And types of chromatography, there are many different ones. So we're just going to mention some of them. So one of the really kind of basic ones would be thin layer chromatography, in which we are separating components of the sample on a thin, almost like a paper-like layer. Well, it's not really paper, but on a layer that is introduced into a solvent, and then the solvent separates our samples on that thin layer. Then we have gas chromatography, in which we have stationary phase and mobile phase, which is gas. So gas is bringing our sample through the stationary phase and enables separation. And then the ones that you're interested the most would be liquid chromatography, in which we have liquid as our mobile phase that is moving and bringing our samples that we want to separate over a stationary phase. And there are many modes of liquid chromatography. Some of them are listed here, and most of them we're gonna look at. So we're gonna talk a little bit more about the ones, not all of the ones, but the ones that are used mostly in biopharma, because not all of those modes are suitable to be used for biopharmaceuticals. So we use some of them. So we'll talk about reverse phase, size exclusion chromatography, ion exchange, and affinity chromatography as well. So in liquid chromatography, this is the part of chromatography that we're gonna concentrate on. We have sample that is dissolved in a solvent. So our mobile phase is a solvent that is flushed through a column packed with lots of tiny solid particles or resin beads. So solvent is our mobile phase, and those solid particles that are packed inside the column are our stationary phase. Different components of the sample will separate due to their either higher affinity for the mobile phase or higher affinity for the stationary phase. So they will either prefer to exit the column pretty quickly following mobile phase, or they will prefer to stay in the column for a little bit longer because they prefer stationary phase. So depending on that, we will be able to separate complex mixtures. Liquid chromatography was defined in early 1900s by a Russian botanist, Mikhail Tsvet. What he actually did, he separated leaf pigments using a solvent and a column that was packed with chalk and alumina. So when he added sample to the column, as solvent carrying those leaf pigments passed through the column, he got different colored bands. So some components were moving faster than the others, and he got really lovely bands. And this is why he called it chromatography, which actually in Greek language it means like writing in color. Fortunately, we don't have such lovely colors in our day-to-day chromatography anymore. Okay, so he basically showed us that the main mechanism of liquid chromatography. Once we put a sample that is dissolved in a solvent onto a column that is packed with tiny particles, sample that is actually composed of a mixture will separate depending on different affinities of the mixture, of the particular substances in the sample towards the stationary phase or the mobile phase. So on this slide here, we see that the yellow substance was attracted least to the column, so it will elute first because it prefers the mobile phase. Then we have red substance that is more attracted to the column, so it will elute later. And then finally we have blue substance that is mostly attracted to the column, so it will elute last because it will spend quite a long time in stationary phase, binding to stationary phase to which it prefers. So each component of the sample can be collected into separate fractions or collection vials if we want to for further analysis. Usually, typically in a QC lab, we're not really interested in that, so different fractions are just analyzed and sent to waste. But during characterization, we might collect each of these samples, each of these fractions, and then analyze them further, maybe using a mass spec, for example. So solvent is continuously flushed through the column to promote movement of the sample. So if we don't put solvent through the column all the time, sample will just stay in the column and won't move through the column. The type of liquid chromatography that we are going to concentrate on is HPLC, so it's high performance liquid chromatography. And in this chromatography, we have our solvent, which is our mobile phase, but we don't have like in the column that I showed you on the previous one, we don't put solvent through and then wait for gravity to move solvent through the column. We have high pressure, so we have high pressure pumps that are used to generate flow through the column. So this is why this is called high performance or high pressure liquid chromatography, and it's one of the most powerful tools really in analytical chemistry because it's relatively easy to use compared to some other most complex methods, and it can give us a huge amount of information about an analyte. So column that we have, or a stationary, in which we have our stationary phase in HPLC, is usually glass or metal tube of sufficient strength to withstand the pressure. So most commonly it's a stainless steel tube that can withstand the high pressure of a HPLC instrument, which is usually around 400 bars and more. So mobile phase will run through the column and permeate the stationary phase that's inside, and stationary phase are small particles, so small solid particles or semi-solid particles or porous particles of very small diameter. So usually in HPLC we have those little particles that are 3 to 5 micrometer in diameter, but they can go as low as 1.7 micrometers in ultra performance liquid chromatography or UPLC, which is like an improved version of HPLC. These particles can be coated with various substances to attract specific type of molecule. So we can have them, for example, coated with a ligand to attract a specific type of molecule, or we can have them charged. We can have them coated with long hydrophobic chains in order to attract hydrophobic regions and bind hydrophobic molecules. So we can vary them depending on what we would like to attach to them. They should have a large surface area that is accessible to mobile phase and then a sample as well, so it allows samples to bind to it. And common stationary phase material is silica. So usually they're made out of silica, which is really a component of sand or quartz, but they can slightly differ. So these are the components of a typical HPLC instrument. And what we are seeing here is our stationary phase. So this is our column that is packed with those small beads of different properties. We have our mobile phase, which is our solvent. Solvent is moved through the stationary phase using a pump or a solvent manager. We have a sample manager or injector. So this is the way that we inject our samples onto a mobile phase that will then bring it through the stationary phase. Our sample will go through the column and stationary phase, and it will get separated inside the column, and then it will exit the column and go through the detector, so we will be able to detect it, visualize it, and we will be able to see peaks of our sample or chromatograms that we can then analyze and make some conclusions about our sample. So separation on the column happens in a way similar to what we've shown already on one of the previous slides. Once we inject a sample onto our column, our sample will be injected as a narrow band, but our sample usually contains a mixture of different substances. And as those substances are traveling through the column, they will start separating based on their affinities towards the stationary phase or the mobile phase. So in this case, our red sample here had the lowest affinity towards stationary phase. So it's going through the column pretty quickly, and it will be the first one that will go through the detector as it elutes. Then it's followed by an orange, purple, green one, and then the last one is the blue sample that has the highest affinity towards the stationary phase. So it is the one that's going to come out of column last, and it will be detected last. So how do we actually see peaks on our chromatograms, or why do we see peaks? So as our sample goes through the column, as our sample is injected, it's a really narrow band, but as it travels through the column, it starts spreading a little bit. And this is because the sample will be most concentrated in the center, and it will start spreading a bit because, like everything else in nature, everything that is concentrated, it kind of tries to go to a less concentrated area. So because of that, our sample will start diffusing inside the column. So as it diffuses a little bit through the column and goes through the column, once it hits the detector, the first part that's going to hit the detector is this really diluted part of the front of the sample, or we call this the fronting end. There is not much protein in there, so it's going to give us a really low response. So this is the bottom, the beginning and bottom of our peak. Then majority of our sample with high concentration hits the detector, and this forms the middle or the center of our peak. And then this is followed by the end of our sample or the tailing end of our sample, which again does not contain a lot of protein, so the response from detector is going to be low, and that's going to form the tailing end of our peak. So we get a peak that's going to be more or less broad depending on how much our sample actually diffused within the column. So if our sample diffused more, our peak will be broader. If we manage to keep our sample in a narrow band, the peak will also be narrow. And then we see our chromatogram. So our chromatogram usually have on the x-axis time, and then on the y-axis it has the response, as we can see here. So we have time in minutes and then some kind of response, and that might be response from a UV detector such as absorbance, or maybe it's a response from a fluorescence detector, so fluorescence intensity. And in this case we have 4 components of the sample that separated, and you can see that they came out of the column at different times. So this first peak came out after around 2.5 minutes, and this is the one that was weakly attached to stationary phase and weakly attracted to stationary phase. And then the last one is the one that was strongly attracted to stationary phase. So it took 25 minutes for that sample to travel through the column and to the detector. So what we can measure is retention time. So we can measure the time that it takes sample to travel from the injection point through the column to detector. So retention time for this peak would be around 2.5 or maybe 3 here. For this one here would be around 25 minutes. So retention time is a quantitative measurement for our sample, and it should stay the same for each sample. So this is how we can determine whether a sample is fine, because we have predetermined retention times, for example, or retention times that need to match our reference standard, so we can compare them. And if they are out of specification, so they don't match our reference standard, we know that something went wrong with our sample, because now it's eluting differently, which means that it has different properties. We can also quantitate chromatograms, so we don't just measure retention time, or we don't measure how long it took sample to travel through the column. That's important, but we can also quantitate the amount of sample. And to do that, we have to what's called integrate our chromatograms, and this is done by a software. And this means is to decide what peaks are actually peaks, where are peaks starting and ending. We need to count the peaks, and we need to determine the area beneath the peaks. So first thing that the software will do, we'll draw a baseline beneath peaks, like the one you can see here. So we have a baseline underneath our peaks. Then the software will determine where to drop lines between the valleys. So where is the beginning and where is the end of the peak? So in this case, you can see that this peak begins here, it stops here. This is where the second one begins and ends, and then the third one begins here and ends here. After we are done with that, we can then number our peaks. And we can see that in this case, we have 3 peaks. Those 3 peaks, if we sum them together, their area is going to be 100%. So those 3 peaks make 100% of the area. And then we can determine relative percentage area of each one of them, depending on that 100%. So relative percentage area of the first one is 20%, of the second one is 10%, and then third one is 70%. So we quantitated peaks, and again, we can compare this to our specifications, or we can compare this to our reference standard and see whether they match, for example, when we are doing this in a QC lab. HPLC method can be run as an isocratic or a gradient method. In isocratic method, we have one mobile phase that is used throughout the run. So throughout the run, we only have one mobile phase. We don't change mobile phase. And you will see we use that in size exclusion chromatography. So we have one mobile phase used from the beginning to the end of the run. Or if we want to improve our resolution and separation, we can do gradient elution. And in this case, we usually have 2 mobile phases that are used, mobile phase A and B, that are mixed before they hit the column. So we can mix different percentages of A and B, and we can change them throughout the run. And this usually greatly improves resolution. So how well we separate samples and how quickly we separate them through the column. So a lot of methods like what you're going to mention, reverse phase for peptide mapping, or ion exchange would use a gradient method. So let's say if we have separation on an isocratic method, you can see that here we managed to separate all of the samples, so A, B, C, D, E, F, but it took almost half an hour to separate all the samples. If we did the same sample, if we did separation on the same sample, but using a gradient method, so it means that we will change composition during separation, we can see that the elution strength of a mobile phase is increased, and we get good separation of all of these samples in much shorter time. So because we started changing the solvents, you can see that starting from 7 minutes, we started increasing the percentage of sodium chloride in this case, and everything eluted a little bit quicker. So our run was actually done after 16 minutes rather than 30 minutes. So gradient methods can greatly improve the speed of your separation. Mobile phases that we use for either gradient or isocratic are usually water, with different percentage of organic solvents that need to be HPLC grade. They need to be of high purity without particulates in them. And then different buffers such as phosphate buffer, or any kind of buffers that are really good in maintaining physiological pH or whatever the pH of the reaction is. And this mobile phase can vary in polarity. So we can use something that is really polar, such as water. So water is really polar molecule. And then we can use less polar molecules as well. So things like acetonitrile, methanol, and ethanol, all the way to hexane. They would be mobile phases that are considered non-polar. They're still polar, but they're less polar than water, which is a really polar molecule. So for example, for a reverse phase gradient method, we might start our separation with 90% water and 10% acetonitrile, and then we can start increasing the percentage of acetonitrile throughout the run. So we end our run with 80% of acetonitrile and 20% water. So that would be a typical gradient method for something like reverse phase HPLC that we are going to talk about more. In most of the cases, what we need to do when we have our mobile phases, we need to filter them and we need to degas them. So degassing is required to remove dissolved oxygen. We don't want bubbles in our mobile phases because that will interfere with separation and might prevent, we won't prevent band spreading. So if we have a gas, that might induce band spreading. So we have spreading of our bands a lot. And buffers often, especially for ultra performance liquid chromatography that is done at extremely high pressure, often they should be filtered through 0.2 micron filter in order to remove any particulates that might damage our column under that high pressure as they are going, as the mobile phase is going through. So that's just an overview. Next thing that we're going to talk about is the theory of chromatographic separation. So how do we determine what is efficient chromatographic separation, what is not efficient, and what columns are efficient and which ones are not efficient? So the most efficient column should produce sharp peaks and minimize band spreading. So the best columns should really stop your sample from diffusing and spreading throughout the column. So your sample should travel as a really sharp, as a really narrow band, so that band will not spread inside the column. And if that happens, and if you remember what we mentioned in previous slides, as it goes through the detector, we're going to get quite a sharp peak. And then that means that we will be able to separate a lot of samples because they are not spreading, bands are not wide, and they will not go into each other. Okay, so tight, narrow bands are produced if our column is uniformly packed. So this is how mechanical separation can introduce it. So our column needs to be packed well, but then also surface chemistry of the packing material. So chemistry of our beads can further influence this. So chemical separation also comes into place. Column efficiency is something that we need to determine, and it's something that we measure in order to see whether our column is efficient and not. And column efficiency is basically talking about what is the maximum number of peaks that can be resolved on a column, or what is the maximum number of sample components, let's say, that we can resolve on a column. And we determine column efficiency by calculating plates, number of plates, or N, or theoretical plates. So this idea originates from petroleum distillation, where they have huge columns in which actual plates exist. So plates exist in that column because they help distillation of petrol. So as petrol is getting distilled, when liquid hits the plate, liquid condenses and different components of petrol can be removed. So the more plates you have in the column, the more different fractions you're going to remove from petrol, and you will have purer petrol. So basically just means the more plates you have in the column, the better efficiency of that column, because you will be able to resolve and remove more impurities from petrol. Inside our columns, we don't actually have plates, so we are just imagining plates. So what we measure the width of the peak that we have on our chromatogram, and then we relate it to the length of the column. In that way, we can determine what is actually the number of plates that we can fit onto a column, and we can determine the width of those plates. And the width of the plates we call HTP, or height equivalent to a theoretical plate. So the more plates we have, that means that we will have more efficient column, because the more plates, the more, theoretically, the more sampled components we can resolve. And also the lower the HTP, the better efficiency, because HTP is height equivalent to a theoretical plate. So if the plate have small height, so if they are thin, that means that we can fit more plates into a column. If HTP is large, that means that height is large, so plates are quite thick, and we will be able to fit less plates. Okay, so the formula from HTP, you can see it here. So N is equal L divided by HTP. So HTP we can get from if we divide L by N, by plate count. Okay, and plate count and HTP are reversely proportional. So the more plates, the lower HTP, and this is what we want. The higher the HTP, the less plates, and this is what we don't want. Okay, so that's plate theory, but that plate theory has three major issues. So it assumes that the equilibration between two phases is infinitely fast. So the last equation assumes that once we put the sample into a column, the sample will straight away bind either to stationary phase or stay in a mobile phase. However, that does not happen. As our sample is traveling through the column, a sample at any point can be in stationary phase or mobile phase. It kind of moves through either or, and it stays in one more or the other more, depending on the affinities. This plate theory also ignores the effect of the velocity of elution on the efficiency and resolution, so it ignores the flow rate. It does not take into account what is the flow rate of our separation, and it also ignores multiple paths that analyte can take through the columns, because our analyte usually does not take a straight path. It usually wiggles through the column. So most realistic description of column efficiency is given by Van Deemter equation. And Van Deemter equation just tells us that HTP, or that column efficiency, depends on three terms. So the first term is called A term, or it's called eddy diffusion. Then we have second term, which is B term, or axial diffusion. And then we have C term, which is resistance to mass transfer. All of these three terms depend on some factors. So as you can see here, A term depends on a diameter of particle, B term depends on speed of mobile phase, and C term depends on both. So we're going to talk about how they depend and what they are actually. So A term, or eddy diffusion, describes different flow paths that are available to molecule as it travels through the column. So as I said, our analyte will not travel through the column in a straight path. It will wiggle through the column, and how much it will move through the column, it will depend on particle size, so only on particle size. If you have smaller particles inside the column, you can pack them much closer together, so you will have less space in between those particles, so sample will be traveling through the column more uniformly. Okay, it's not going to be able to wiggle because we packed our column really tightly. If the column particles are really large, we're going to have a lot of space in between those particles, and our sample can really go through many different pathways. So the smaller the particles, the better. Axial diffusion is describing how much our analyte diffuses as it travels through the column. And it's inversely proportional to velocity. So if we do our separation using really slow flow rate, analyte will spend longer in the column and we will give it longer time to spread. And then if it spreads, we're going to have wider bands, which we don't want. If we go too quick, then our sample will actually fly through the column too quickly and it's not going to be able to separate within the column. So we need to find a good flow that is not too slow or too quick, especially not too slow because we are diffusing our sample too much. And then C term is resistant to mass transfer. So it describes the effect of speed as the analyte equilibrates between mobile and stationary phase. So as I said, when we add our analyte to our column, our analyte will be moving all the time between mobile and stationary phase, in and out of mobile phase and onto the stationary phase. This is related to velocity because if we have too high speed, we will not allow our sample to interact with stationary or mobile phase. We will just push it really quickly through with the mobile phase. If we do it too slowly, then we're going to get a lot of spreading. This also depends on particle size because the smaller particles we have, the larger the surface will be. The larger the surface, we will allow for more mass transfer because we will give our analyte more surface to attach if it wants to attach. So C term depends on velocity and particle size. So if we get them all together, we get one Dimeter equation, and we got a graph that allows us to find an optimum velocity for a certain analysis. So here we have plate height or HTP, and you can see that HTP is not really going to be affected by A because A is not really affected by speed, so it doesn't matter. It only depends on the particle size, so the smaller the particle size, the better. But you can see here, if we have really low B, we're going to have really high HTP, and this is what we don't want. We want low HTP because at low speed, B will really be dominant. At low speed, we will have a lot of diffusion of our sample inside the column. C, however, it's going to be really dominant at high speed. So again, at high speed, we're going to get really high HTP, which we don't want. And this is because at high speed, we are not allowing for mass transfer because we are forcing our sample too quickly through the column. So there is a sweet spot here where you have the lowest influence from B and from C, and if you can get a small particle size, you get the best HTP or the best efficiency of the column. So basically, reducing A, B, and C term will lead to decrease in HTP, so that peak width, and increase plate count, because if we decrease HTP, we increase plate count, so that means that our column is more efficient. And then we will also increase resolution, so how well we are separating between those complex samples. There are many other factors that also affect this, but this is the main point of separation. So we want to have nice, sharp peaks that are quite symmetrical. So it means that they are equal on both sides, but what can often happen is fronting and tailing peaks. So if we get peaks that are not symmetrical, that are either tailing or fronting, that is an indication that the conditions in our HPL methods are suboptimal. And maybe our mobile phase is not right, our column packing is not right, or we have blockages inside the column. So tailing just means that this tail end of the sample will spread. So if we look at how sample is traveling through, we can see that the majority of sample travels out, meets the detector, and then we have a lot of sample at the tailing end that will trickle through the detector. So this way we have this long end, tailing end. Fronting happens if it's other way around. So we have this first our sample is quite diluted at the front, so it trickles to the detector. So we have a really long front, and then the majority of the sample comes out. So fronting and tailing are caused with something going wrong with our HPLC system. So tailing can be caused if we have blockages at column frits. So column frits are bed supports. So bed supports on the inlet and outlet of our column that allow stationary phase to stay within the column. So they stop stationary phase for exiting. And they can get blocked. And if they get blocked, our sample will not be able to go into the column. So some sample will be able to pass through, but a lot of it will won't, so it will tail afterwards. It can happen because of secondary interaction between analytes and the columns. So sometimes we want separation, for example, just based on size, but maybe because of either a wrong mobile phase or samples being charged or something like that, we might get a bit of separation due to charge as well, even though we just want size. So we get this secondary interaction. So tailing usually informs us of some chemical problems with separation. Fronting is usually caused by a column void. So if we have a lot of liquid on top of the column, or void inside between the beads, that means that some of the sample will go through the column pretty quickly, but a lot of it will come afterwards. And this is usually a sign of physical or mechanical problem. I think we have it here as well. So you can see normal flow here, and you can see disturbance here because of partially blocked frit, so tailing. So here it's partially blocked, so some of the sample goes through, some of it will go through later. And on C we see fronting because we have column void. So you can see that column is not equally packed, and we have almost like a hole here. So some of the sample will just quickly fly through, and we're going to get that fronting, and then the rest of the sample will follow through. Okay, so the last few slides are going to talk a little bit about main HPLC methods that we use for the separation of biopharmaceuticals. So usually in biopharma, we separate our chemicals or our substances based on these few different characteristics. So we can separate them based on polarity, based on their electrical charge, on their molecular size, or biological affinity. And then we have those main methods. So this first one, normal phase chromatography, this separates samples based on their hydrophilicity, so how much they like water. But this is not used as much in biopharma. So this is more a method that is used for small molecule drugs. What we use more is reversed phase chromatography, so opposite of normal phase. It's the one that separates our sample based on their hydrophobic properties or hydrophobicity. Affinity chromatography separation of sample happens because of biorecognition. So we have a ligand that specifically recognizes and binds our molecule of interest. In size exclusion chromatography, we separate our sample based on size. In ion exchange, we are separating it based on charge. And then we have a version of normal phase that is used in biopharma, which is called hydrophilic interaction chromatography, or HILIC, that separates samples based on hydrophilicity. Just slightly different than normal phase, uses slightly kind of more let's say, gentle environment for the protein to be separated in. So what does it mean when we talk about polarity of our sample? So some components are polar, some molecules are polar, some molecules are non-polar. That will depend on the separation of charge across the molecule due to an even sharing of valence electrons between atoms. So water is quite a polar compound because we have uneven sharing of electrons between atoms. So oxygen atoms here are positively charged, so hydrogen ions are positively charged, so they will attract those electrons more than an oxygen. So we will have polarity, so unequal sharing of electrons within a molecule. In ethane here, this is a non-polar molecule because all the electrons are shared equally, so there is none of that non-equal sharing of So polar compounds would be, for example, water. Non-polar compounds, they're more hydrophobic. They're more kind of like water hating, while polar compounds are hydrophilic. Would be things like chloroform or things like oils, things like fats, or everything that you can imagine that does not mix with water would be quite hydrophobic. So in reversed phase chromatography, we separate molecules based on this hydrophobicity. And the chemical attraction that is involved is hydrophobic interaction. So what we have in reverse phase chromatography, we have a stationary phase that contains silica particles that have non-polar coating or hydrophobic coating. And we have a mobile phase, which is actually a gradient that starts from mostly aqueous and moves to mostly organic as our separation occurs. Hydrophobic compounds are attracted at the beginning of the separation. Hydrophobic compounds, they don't like mobile phases because it's going to be mostly aqueous at the beginning. So they will be attracted to the non-polar stationary phase. So they will all bind, and then they will elute as we start increasing organic, depending on how much they prefer organic phase. So they will all elute at different times. Polar compounds, on the other hand, will be attracted to the polar mobile phase, and they will move faster through the column, and that will create separation. So this stationary phase, we said that it has hydrophobic coating. It's actually made up of hydrophobic alkyl chains that interact with the analyte. So they're really long chains that can have either kind of 4 carbon, 8 or 18. So they can be short like C4 or much longer like C18. C4 is usually used to separate proteins, while C18 is usually used for peptides and small molecules. Proteins are quite hydrophobic, so we give them slightly less hydrophobic stationary phase because they don't need it. They're already hydrophobic, so they will bind to it. Peptides are much smaller, so we give them this longer stationary phase to encourage them to bind. If we use this C18 stationary phase with protein, this is super hydrophobic. Proteins are really hydrophobic. They will just bind to it so much that we would not be able to detach them from the stationary phase anymore, no matter what we use. Okay, so we will destroy them before we would get them out. So for large things like proteins, we use less polar alkyl chains, and for something that is not as hydrophobic, such as small peptides and small molecules, we use C18 stationary. It's a longer chain. Reverse phase chromatography is used for, it can be used for protein quantitation, for separation of complex protein mixtures, for detection of isoforms, and what's really important is for peptide mapping. So to actually see the fingerprint of our protein, whether we have the right protein, whether we have the right amino acids. So peptide mapping is done by digesting a protein with an enzyme, with a protease, usually trypsin, to cut it into peptides. So trypsin will cut our protein into smaller peptides in the way that we cannot know, because we know where trypsin will cut. And this will be used to produce unique fingerprint of our individual protein. So we denature our protein to allow protease to go close to the protein. Protease then cuts protein into small peptides, and then we analyze those peptides. And they're all cut in the same way, so we know where trypsin will cut the protein. Then we get peptide maps, so we can see all the peptides, and we can put them back together, so like almost like a puzzle, to determine whether we actually have the right protein. So for example, if we peptide map a BSA, we can see whether something is wrong. So we have here in blue control BSA, and then we have BSA in which we what happened is some of our asparagine amino acids got deamidated. And you can see that these two, when we overlay them, we see differences between the control and deamidation. So our protein actually changed. So we can see smaller changes on amino acid level when we chop it up into smaller pieces. And lower here we have control again in blue, and then the oxidized form of BSA. So some amino acids got oxidized, and we can see again we are not seeing the overlay, so we would know that something is wrong with the protein. Amino acids are slightly different. Then we have ion exchange chromatography. Here we have separation of molecules based on their charge. And stationary phase that we have are particles, usually agarose, to which positive or negative charge has been applied. And our mobile phase is usually aqueous buffer. And we can change the pH or we can change the concentration of that buffer to help binding and elution. So charged compounds with the opposite charge to stationary phase will be attracted to the particle surface and be retained. So if we have positively charged stationary phase, that will attract negatively charged compounds. So in ion exchange chromatography, we can have either we can have beads that are either anion exchanger or cation exchanger. So anion exchanger, they are positively charged because they bind negatively charged anions, and cation exchangers are negatively charged because they bind positively charged analytes or cations. Okay, so separation depends on the adsorption of charged solute to immobilized ion exchange groups of opposite charge, as I said, on our resin beads. Ion exchange separation consists of 4 steps. So first step of separation mechanism is equilibration. So during equilibration, we need to condition column with the starting conditions so that we prepare it for the sample application. So we need to make sure that all stationary phase groups are associated with exchangeable counter ions. Then we add our sample. So sample is loaded at the same pH and ionic strength as the initial conditions for equilibration. And then appropriately charged proteins will bind while everything else will be washed out. And then we have elution. So we are eluting retained samples by altering protein conditions. So we change salt gradient. So either pH or we change the concentration of salts. And we elute stuff out, and then we have to do regeneration. So we have to wash column with harsh conditions to completely make sure that everything is detached from the column, and we return column to its original conditions. And then we can proceed to equilibration. So elution can happen by adjusting the pH of the mobile phase to change the charge. So if something was positively charged and was bound to negatively charged beads, we can elute by changing pH and making that now negatively charged, so it will come out, or we can change the charge of the stationary phase, for example. We can also add ions to the mobile phase, so change the conductivity or concentration of our buffer, and now these ions will displace the compounds from stationary phase. These ions from the mobile phase will actually have higher affinity to stationary phase, and they will push everything that's bound to stationary phase off the column. So we can vary those. And we can vary those because if we change the pH of the environment, we change the pH of our protein. As you probably know, every protein has a pI, or a pH at which it's going to be neutral. So, for example, this protein here is going to be net neutral or pH 5.3. So if we know pI of the protein, we know then if we put that protein into a lower pH than its pI, it will become net positive. And if we put it into an environment with higher pH than its pI, so for example 5.8, it will be net negative. So by knowing that, we can change the pH of whatever we have inside the column. IEX is used to examine charge variants of different charge isoforms of our drug, and to monitor chemical changes to protein during processing. So if things like oxidation happen or deamidation happen, they would lead to chemical change. So that will change the charge of the protein, and then that will change retention time. So we will get different retention times, and we will know that something was wrong with our protein. So you can see here we have charge variants identified using IEX. So we always have, when we analyze it, we always have our main peak, and then everything that came out before main peak is mostly negatively charged or acidic. In this case, if we have resin that is actually a cation exchanger. And then on the right side, we have stuff that came out that are more positively charged or basic. So basically, more negatively charged came out first because they were not attracted to the resin. Then our main peak, so our main isoform came out, and then our more kind of positively charged isoforms came out because they were quite attracted to this resin, which was negatively charged. Then we have size exclusion chromatography. In size exclusion chromatography, we separate molecules based on size, and there is no chemical attraction involved. Our stationary phase are silica particles which have pores of defined size, and our mobile phase is aqueous buffer that we don't change throughout the run. This aqueous buffer is only here to control pH and ionic strength of protein, and this is isocratic method. Small proteins will penetrate pores in the column, and they will progress through the column really slowly, while large proteins cannot go through the pores of the stationary phase, and they will go through the column faster. So this is how we will get separation. So small molecules, these purple ones, will be able to go through the pores of the column, so they will go in and out of the pores of the stationary phase. It will take them a long time to come out of the column, so that means they will end up more right on our graph. While large molecules can't go through the pores, they go around the resin beads and come out really quickly. So they enter a few pores and travel rapidly, and they are eluted sooner. So whatever comes out first, the first peaks that we see are larger and then followed by smaller and smaller particles. So you can see here we have a protein mix. We have 3 different components that eluted in different times. So the first one, protein 1, eluted quickest because it's the largest one. It has size of 669 kilodaltons. Protein 2 is 80 kilodaltons, so eluted afterwards, so it's smaller, so it stayed longer in the column. And then protein 3 has its size of 29 kilodaltons, so it's the smallest one, so it stayed in the column the longest. So you can see that it took it 3 minutes to get eluted. Size exclusion chromatography is currently the primary method to detect aggregates. So if we have any aggregates inside our biopharmaceutical product, we want our biopharmaceutical product to be just as one molecule inside. So we have only one molecule that are floating, and we don't want our biopharmaceuticals to start binding to each other. So we get 2 molecules sticking together, or 3 or more, because they can be a huge issue, a huge safety issue for patients and a problem in production. So SCC is used because it's quite fast. Sample analysis takes approximately, well, it can be even less, so it's only like a few minutes to analyze one sample. It's quite efficient, quantitative, and it has quite high precision. So it detects aggregates. So what we always want to see are monomers. Some of our components will dimerize, so we will sometimes have 2 molecules of biopharmaceutical binding together, but we don't want high percentage of dimers. And what we don't want to see are these high molecular weight aggregates, because they're a safety issue. If they are injected into patients, they cause immune reaction. So we have to test our biopharmaceutical to determine the amounts of dimers and high molecular weight aggregates, and they have to be really low. So that is all. There is some further reading on your Moodle page, so you can have a little bit of a look later. And before I go, the final thing that I want to go through is your final LAQ exam overview or long answer question.